So how do these plants obtain this light energy? What structures do they use? What mechanisms do they use to make sure that they can trap this sunlight energy? We have what you call growth movements. And in those growth movements, in coordination, we talked about them. We said that we have what we call trophic movements or growth movements, where plants grow towards or away from a particular stimulus. And in this case, we're talking about phototropism, that the plant will always grow to where the light is. And that is an adaptation to ensuring that they actually obtain that light that they need. Etiolation is if you have a plant that is growing in shade, under the shade, okay, growing under the shade, it means that it does not get exposed to sunlight, and that's a problem. So what these plants do is that the shoot grows or elongates very rapidly such that it can reach where there is light. So that is what you call etiolation. And then we have the mosaic leaf arrangement, that's such that you don't want to have the leaves of the same plant covering other leaves. So that means that the leaves have to be arranged that they do not cover or they do not inhibit the exposure of other leaves from getting sunlight. So that's what we talk about there. So it prevents overlapping and also prevents shedding of neighboring leaves. The leaves are usually broad. That means that they have a larger surface area for absorption of sunlight. And then others are thin. That means that the distance that light will need to travel is actually minimal, and that means that they are very efficient in absorption of light. The cuticle that prevents the evaporation of water is also very important in allowing sunlight to pass through, because it is actually transparent. Imagine if that cuticle is opaque. That means that light will not go through, and that means that the chloroplasts will not be able to get that light. And then the cells of the palisade mesophyll layer are actually closely packed. That means you have very many chloroplasts in there. Remember, the palisade cells contain chloroplasts, and that means that if you have many of these palisade cells, and that means that you have many or numerous chloroplasts in there. These chloroplasts are actually not static, and they can actually move. That means that they can actually reposition to the direction of light. So that's the process that we call cyclosis, okay, process of chloroplast movement within the palisade cells. The ordered arrangement of chlorophyll is also another important adaptation that we have for plants being able to maximize light absorption. Some plants actually have multiple layers of the palisade mesophyll in the palisade mesophyll, and that means that they have very many chloroplasts in the upper palisade mesophyll layers, and that is important in maximizing light absorption. And then, of course, the process of photosynthesis requires gases. It actually requires carbon dioxide and will leave out oxygen. And that means that there should be efficient mechanisms to ensure that this gaseous exchange actually takes place efficiently. You don't want to have a buildup of these gases without being used. So numerous stomata, the openings mainly on the lower epidermis, and help in this role. These stomata have what we call guard cells that can enable the stomata to open or even close. The spongiomesophyll cells are actually irregular, and that means that it creates spaces that allow for movement of gases very fast compared to what happens in the pulsed layer. And then we talk about transportation of the products of photosynthesis. Of course, you'll have glucose as one of the products of photosynthesis, and this has to be moved to different places where it is actually required. And the role played by the vascular bundle, where you have the xylem and the phloem, becomes very important. So the phloem will transport the sugars, that is usually, of course, when you talk about photosynthesis, we talk about the synthesis of glucose, but the transportation process involves sucrose. Sucrose is the transport form of the carbohydrates that we have in plants. And then the xylem, very important in transporting water and mineral salts. So the leaves have the vascular bundle that consists of the xylem and the phloem. And then, of course, that is usually, if you get the structure, you remember the structure of the leaf, where we have the midrib, but also the midrib has branchings or networks that branch away from it, and these still have cells that can conduct these, that can transport these sucrose and water and the amino salts. And for example, the point is that you have these fines, that is, means very small veins that branch away from the central midrib, and these have branchings that are very close to the xylem vessels, and that is very efficient for transportation of water and amino salts, and then for the phloem sieve tubes for transportation of the sugars. Remember, since the leaves are the ones that make the sugars, these sugars have to be moved away to other parts where they have to be used. We often call them the sink. When we look at transport, we shall look at the transport of organic compounds in plants. We shall talk about the sink and the source. The source is where we have the different, you have the synthesis of these organic compounds. Basically, the leaves are the source, because the leaves are the ones that carry out photosynthesis majorly. And then for the sink is where you have these sugars or organic compounds being used for different processes. So that's that. So we earlier on emphasized that the chloroplasts are the ones that carry out the process of photosynthesis, because they have, they have the pigment that can trap sunlight, and that pigment is what we call chlorophyll. These chloroplasts come in different shapes, so you will not have the chloroplasts having the same shape throughout. So usually when we are drawing the structure of the chloroplast, we actually use the biconcave convex. We use the biconvex shape, okay? But this is not always to mean that it has to be of this shape. Yeah? In the organisms such as the algae, you can have the chloroplast be filamentous or even spherical or ovoid in other species. They are very, very tiny. You can't basically see them with your naked eye. So when you're drawing the structure of the chloroplast, ensure that you have the two membranes. Chloroplasts are double membrane organelles. Okay, they have two membranes, the outer membrane and the inner membrane. That's the first thing you start with. The next thing that you go to is ensuring that you have the organelles that the parts that are inside the chloroplast. You have starch granules, you have lipid granules, you have the ribosomes, you have DNA, and then the distinct features are actually the thylakoid membranes, which actually get packed in two groups that we call the granum. Okay, that we call the granum. Here, if you see this is the granum. Okay, so they are basically the thylakoid that are packed. So one of these is what you call the thylakoid. So what connects one granum to the other is what we call the intergranular lamella. So these granum are actually connected to one another through the intergranular lamella. That the chloroplasts actually have DNA, which is not indicated in this structure. It also means that they are semi-autonomous. So we have what we call chloroplast DNA. Okay. So inside the chloroplast, we have what we call the stroma. Okay. Stroma is where we have all the other elements, the granum, the thylakoid, the starch grain, lipid globule, ribosomes being in there. This granum is very important because it is involved in exchange of materials and synthesis of fatty acids, lipids, and carotenoids. Okay. So the thylakoid are the ones that are flattened and they are interconnected. This is where you have this pigment called chlorophyll. Okay. So you have the fact that you have the thylakoid being packed into granum. This actually increases the concentration of chlorophyll. Okay, because the chlorophyll is actually on this granum. So it increases the fact that you actually see that you will have processes of photosynthesis happening on the thylakoid membrane. So the fact that we have these stacked thylakoids, it means it increases the surface area for photosynthesis. And the stroma, like I already explained, is that jelly-like matrix where you have all these other structures in there. And, the enzymes, the ribosomes, the chloroplast DNA, starch granules, lipid globules are all there. So how is the chloroplast adapted for photosynthesis? It has a semi-permeable membrane. That means that it can control what enters and what gets out of it. Okay, and this is very important, regulating what happens inside the chloroplast. The thylakoid membranes, the fact that they are actually very extensive and also have enzymes and also chlorophyll that is very important for the process of photosynthesis. We have enzymes in the stroma. We have the chlorophyll that is also there in the chloroplast. The fact that the chloroplasts have their own DNA means that they can control their own affairs. That's what we mean by semi-autonomous. So they don't have to wait for the cell nucleus to basically direct on what needs to be synthesized and what enzymes need to be made. Okay? The small intermembrane space, that space that is between the inner and the outer membrane, is very good for establishing the proton gradient, which we shall see that is actually very important in the synthesis of ATP in the process of protein synthesis. So that marks the end of this presentation. We have noted that the leaves are mainly the parts of the plant that are actually responsible for photosynthesis, but you can also have other green parts of the plant carrying out photosynthesis. The green plants that we see, or the green parts of the plant that we see, are actually because of chlorophyll. Chlorophyll is a pigment that actually traps the sunlight, and this is actually located in chloroplast. So we can basically say that the chloroplast are the site for photosynthesis.